Hello, welcome to another screencast. This time we'll be talking about the PCR, or the polymerase chain reaction. So before you do this, you should take a second and review here what you know about DNA replication. So the enzymes and the steps and the details involved with DNA replication are critically important to the polymerase chain reaction. Following that, you should be able to, at the end of this here, describe what PCR stands for, at a minimum, the purpose of PCR, and then the materials needed for PCR. But then really down here, you're trying to explain how PCR works, what the results of the PCR are going to look like, and then most importantly, ways in which PCR can be used, right? So what kind of questions can PCR be used to help answer, and then how do you actually interpret the results of the PCR reaction? So let's get started. Again, just a quick review of DNA replication here, where this DNA polymerase is making a complementary strand of DNA here in light blue to this template strand of DNA here. This all started off by using, of course, a primer here that DNA polymerase can then add to this 3' end, right? So the PCR is based off of this replication mechanism, right? So again, the first part of the replication mechanism, remembering here, is that a primer is going to be laid down. Yeah. So this primer is complementary to a specific sequence of nucleotides in the DNA, right? So this is sort of something that's important, right? That the primer is a specific sequence of, in the case of replication, RNA, right? But it could also be ssDNA, single-stranded DNA, right? So this primer that we're going to use in PCR is known, is of a known sequence. Okay, so that primer is a known sequence. Those primers are described as being specific, right? And when the primers bind to the DNA, we say that the primers are annealing. This is annealing is also what they really mean here is that those primers are going to hydrogen bond, right? So again, we're going to see this annealing here, bonding, hydrogen bonding, the binding of the primers to a specific DNA sequence. Okay? So again, this PCR is based on something that's normally naturally happening in cells, and that is the DNA replication. So here we go for the PCR. The PCR stands, for the polymerase. This is the P. Polymerase chain reaction. Chain reaction. So a chain reaction is once you get it started, it keeps going and going and going and going. And the polymerase here that they're going to refer to is a DNA polymerase, right? And it's actually a special DNA polymerase, which we'll talk about in just a little bit. So to do the PCR, I will back up a second. So what is the purpose of PCR? The purpose of PCR, as the title implies here, amplifies target sequence DNA, right? So by amplify, we mean that the PCR is a way to make many copies of a specific DNA sequence, right? So making many copies, right? Amplifying. Another way that you also might hear this referred to is that you are cloning, right? Making lots and lots of copies of the exact same sequence. Right? That is the whole point of PCR. Right? Now, where is this happening? This is happening in vitro, meaning in glass. Right? That is not in a cell, but instead of glass, we're going to be using little teeny tiny plastic microcentrifuge tubes. Right? So, in glass, plastic, whatever, same difference. So, what do we need? You need a DNA polymerase. Right? You need primers, and again, these primers are single-stranded DNA primers. They are known sequences. Right? They are short, right? So they are oligonucleotide sequences. Right? They are complementary to some sort of known sequence that you're trying to amplify. Right? And you also need then a supply of nucleotides over here. So this D, this stands for that you have deoxynucleotides, right? So deoxy ATPs, deoxy CTPs, GTPs, etc. So these are the these are nucleotides that you're going to use to then amplify. So what happens in the PCR is that we know that in DNA replication, this double-stranded DNA molecule here will first be separated by helicase. Right? But now in vitro, what we do is that you actually heat it up. Right? So heating it up, it denatures and it breaks the hydrogen bond. So this heating, right, breaks the hydrogen bonds between the DNA fragments here. And you're going to separate the DNA strands into single-stranded DNAs. Then you cool this back down to allow the primers to hydrogen bond, right? And so you heat it, you cool it down. Here you see the primers bonding, right? And here's a special note. These primers are only going to bond if this sequence right here in the DNA is present, that is complementary to the primers, right? So this is kind of a key point right here. Right? Primers only bond if the DNA sequence that is complementary to the primers is present in your sample. So if the sequence is not present, then the primers are not going to bind. Okay, but let's just say that the primers bind here, and this provides a 3' end to add to. Right? And so now the DNA polymerase that's in there, once these primers bind, the DNA polymerase adds nucleotides to the 3' end of each primer, right? And you get then a new double-stranded DNA, and this holds with the semi-conservative model. So this highlights the semi-conservative model, or it gives you an opportunity to review the semi-conservative model. And so now I have 2 copies, one here and one here, of this initial target sequence up here. And that's after 1 cycle. Now I'm going to cycle this again, and by cycle I mean, so the cycle part of this means heat, cool, right, and then polymerize, sorry. Heat, cool, polymerize. Heat, cool, polymerize, heat, cool, polymerize, right, so on and so forth. So after the second cycle, right, and you can follow this here. So here is the original primer. Here's the new strand. It was complementary, right? And now I'm going to heat that up. That's going to break this into single strand, and I'm going to get this. So in this image right here, this part right down here, they're skipping showing you this part. Right? And you do this over and over and over again, and each cycle doubles the target sequence that you are looking for. Okay, muchly encouraging you to watch these animations. They do a much better job animating it than I'm talking about statically here. Okay, but again, this is the way that we are going to amplify or clone. We have lots of copies now of this target sequence. Here's another visual to take a look at. So again, PCR is going to amplify target DNA sequences in vitro. So if we start with our target sequence right here, after cycle 1 we get 2 copies. After cycle 2 we're going to get 1, 2, 3, 4 copies. Cycle 3 we should have 8. 1, 2, 3, 4, 5, 6, 7, 8. Right? And you see that after this third cycle we start amplifying just this target sequence. These other sequences up here, these other fragments up here, right, will all have sort of these overhanging areas that are not part of the target sequence that we're looking for, and that's okay, right? But that's just the mechanism here. You get these you cannot not get these overhanging sequences here, but that's after 3 cycles. So after 4 cycles, you see it now, we're starting to get lots and lots of copies, way more copies here of just the target sequence that we're looking for, and the ratio of the target sequence we're looking for to all these other fragments that have these overhang areas here, right, is becoming greater and greater after cycle 5. Right? Cycle 6, again, same concept here. As you have lots, these are all fragments. Whoops, sorry. Those are all fragments of the target sequence. That's after 6. Now PCR is done, upwards of 30 to 60 cycles happening here. And so after about 20 cycles, look, the precise DNA length that you have is well over 1 million copies, right? The intermediate ones, these are like the intermediate ones here, and then that template DNA, you have one right there. So, well, remember that all this is happening inside one of these microcentrifuge tubes, right? So you can read that. That describes what we're talking about there. Now that's happening in a tube, but to see if the PCR actually worked, you need to use electrophoresis to actually separate that. So PCR results here are visualized using gel electrophoresis, because remember, now you have, let's forget what this says right here, but in this tube, right, you would have, go back to this other image, right, in that tube, you would have this, right? This whole mixture right here would be in one of those microcentrifuge tubes. This is supposed to be the tube here. You take this out and load that into the gel, right? So gels separate based on fragment size, right? And so I'll skip that. And this is the result. This would be the result of a PCR gel. This is what PCR gels look like. Now, I'm going to just point out here that the PCR gels often look like restriction digest gels. So you need to read the captions down here to tell you, to let you know, like, okay, this is the result of a PCR gel, not, or so this is following PCR amplification. So this is not an RFLP, a restriction fragment like polymorphism result here. Okay. So I think I'm actually going to stop at this point, and then I'll do another little brief screencast for the application of this. Right. So at this point, you should be able to here describe first what PCR stands for, the purpose of PCR, your materials that you need, explaining how PCR works, and then what the results of that PCR are going to look like. And I'll do another screencast for this down here. Okay. And again, go and watch those animations. That is going to be, I think, a little bit more helpful. Okay, questions, bring them to class, and watch the next screencast in this series. Thanks.